Hiya 12. This is just a bit of an explanation for question 5A on the Transport in Animals exam style questions. So just to go through from the start, the first question is fairly straightforward, and we've seen this in lots of exam questions over and over again. So really typical marking points: explain the need for transport systems in multicellular animals. So remember, if you're a multicellular animal, you're much more likely to have, relatively speaking, a higher metabolic rate, okay? Particularly if you're an organism that's going to maintain its body temperature like a mammal, okay? So as a result of that higher metabolic rate, you're going to have a really big demand by those cells for a really efficient supply of things like glucose and oxygen for cellular respiration. But these organisms are also very busy metabolically, making new molecules, breaking new molecules down, cells communicating with one another. So they need a really efficient supply of things like amino acids. They need hormones transporting around. So you're going to need a really efficient transport system for that. Now, if you were a small single-celled organism, you'd be able to rely on substances just simply diffusing in from the external environment. But obviously, if you've got a very high metabolic demand in your cells, and bear in mind we could be talking 50 trillion cells in case of a human, then actually just to rely on diffusion from an external surface alone would be just way too slow to meet the needs required. The other thing you've got to remember to talk about in an exam question is that large multicellular organisms have a small surface area to volume ratio. So again, diffusion is just not going to be possible. We're going to be talking about much longer distances. Even if you think about the exchange surface, an exchange surface in the lung is still a long way for substances to be transported to all the respiring cells of the body. So we need an effective transport system to make the link there. Hopefully that question was quite straightforward, and the marking points make it really clear what to put for that answer. The second question, part B, is something that a few people have asked about, so I just want to go through this one. So it says the diagram show the sequence and duration of events in one cardiac cycle in the mammalian heart. Now, it says on here the letters P and Q refer to two different types of heart chamber. Well, we know there are only two types of heart chamber, so one of these has got to be the atria and one of them's got to be the ventricle. It also says the letters V and W, X, Y, and Z refer to periods of contraction or relaxation. Remember, contraction events we refer to as systole or systole, relaxation, diastole, okay? And so it says identify the type of chamber labelled P and describe the stages V and W of the cardiac cycle. So here we can see V, and if we link it up to the diagram above, and we've got to use the diagram a little bit to help us here, we've got to work out, right, what must P be? So if we're having a look, we can see that actually in the diagram, these chambers here are in a different state than they are in this diagram here and this diagram here. Here we can say that they're very much contracted, and there's a smaller volume inside. Here we can see them both in relaxation. So that tells us that actually in this bit here, we can see these chambers here, the atria, are contracting, whereas here they are both in relaxation. So if we marry that to the diagram below, yeah, here we can see we've got one contraction or relaxation event, we don't know yet, which lasts a short amount of time, i.e. this bit here, and then a relaxation event which lasts for this period of time shown in this diagram and for this period of time shown in this diagram here. So that means P has to be the atria, because atria are contracting here during this between 0 and 0.1 seconds. And then here we can see the relaxation phase between 0.1 and 0.8 seconds. And that kind of makes sense with what we know about the cardiac cycle. It then says, describe what's occurring in V and W. So all you'd need to do to pick up the marks would be that in V, it's contracting or undergoing systole, and then in W, it's undergoing diastole. So that's what we can see here. Here we can see in the ventricle, and it doesn't ask us to talk about this yet, but here we can see in the ventricle, we've got almost three separate events. We've got either a contraction or a relaxation, a contraction or a relaxation, a contraction or a relaxation. And if we look at then referring back up to the diagrams above, we can see, right, looking at the ventricles, yeah, they're very much in a much more relaxed and filling stage in this diagram here and this diagram here. And actually in this central diagram, you can see them in the contraction phase. So then we go back and fit it to the stage here. And it says, for the second question, part 2, state the name given to stage Y. So if we have a look at stage Y here, referring to the diagram above, yep, they're both contracted, those ventricles, so it must be ventricular systole or ventricular systole. Next question is about applying your knowledge of the cardiac cycle. And it says, describe the mechanism that allows the Y stage in chamber Q. So it's talking about the contraction stage in the ventricle, because we've already got our heads around that. So the contraction bit of the ventricles to occur shortly after the contraction stage in the atria. Here we can see the contraction stage in the atria, and contraction in the ventricle appears afterwards. So we need to go back and think, right, what is it that allows the atria to contract first and then the ventricle to contract second? Why don't they kind of contract simultaneously? And what we've got to remind ourselves, and hopefully you watched the video that covered the cardiac cycle. It was quite a nice explanation, and particularly the one on where it related it to the ECGs. You could see that, obviously we know to initiate atrial contraction, the sinoatrial node sends out a wave of excitation across these atria here, which therefore make the atria contract. That wave of excitation or depolarization, and it doesn't matter which one you call it, is prevented from spreading right over the external surface of the ventricles because of a band of non-conducting tissue that lies across here. Okay, now those collagen fibers in that non-conducting tissue prevent that spread of electrical excitation. What happens is that excitation gets picked up by the atrioventricular node in this little region here, and we talked about it in class as kind of catching the impulse and giving a delay. Those impulses are then channeled down Purkinje fibers within the bundle of His, so down, which lies within this central septum region, the actual physical bit of tissue between each of the ventricles. They're channeled down the bottom of here to the apex, the tip of the heart, and then the excitation spreads upwards up the sides of the ventricle, initiating the contraction. But obviously that initiation of the excitation passing up here to cause contraction doesn't happen until you've actually finished contracting your atria, and they've then gone into this kind of diastole relaxed state. So that's what you'd need to be saying for those bits at the end. The final question is asking you to describe how a named piece of apparatus can be used to measure the length of stages V and Y. So that's the concept of relating the actual ECG machine, and obviously you'd get credit for actually mentioning the piece of apparatus and how very simply it works. So you put electrodes on the skin of the chest, and you get the sort of electrical disturbance shown on a trace, and we know that initial little P wave. So in our trace, the little P wave is to do with atrial contraction. That QRS wave, the larger one, is to do with contraction of the ventricles. And what you would be able to do is then, in order to relate it to the length of the stages, well, obviously the distance on the trace paper indicates a relative amount of time, so you'd then be able to record it there. So that's what you needed to say there. Hopefully this has been helpful for you. Just send me any more questions and I'll try and help. Okay, take care.